Hey everyone, my name is Marlene Reyes and I am a current senior. So, the study that I looked at was conducted in 2021 and it is titled Pomegranate Juice Moderates Anxiety and Depression-like Behaviors in Aluminum Trichloride Treated Male Mice. So, initially, I was really interested in doing research um, on mental health in mice in general. So, while looking for different studies, a lot of the papers that I found talked about pomegranate juice a lot, or just the fruit in general was mentioned quite a bit. So, I was really confused at first. I was just like, what does pomegranate juice or pomegranate in general have to do with it, and how does it help prevent some of the negative effects of these mental health issues in the mice? This study in particular really stood out to me because of the aluminum trichloride. I did not know that aluminum or exposure to aluminum trichloride could lead to anxiety and depression in mice. But after some research and after looking at different papers and uh, different sources, I did find that recently it's been discovered that many metals are linked, or some metals are linked to many neurological pathologies. So, a lot of researchers are on the hunt to find different methods that can alleviate the effects of, the negative effects of the exposure to these metals. So, just aluminum in particular is very ubiquitous, so meaning that it's found everywhere, and it does have a great impact on environmental pollution as well as food contamination. So the exposure of it just becomes inevitable. So specifically, aluminum trichloride exposure was proven to encourage the apoptotic process in the hippocampus, which then leads to behavioral deficits and therefore inducing anxiety and depression. So, a lot of modern studies have been trying to find different uh, different ways to control this exposure, how to moderate it. And pomegranate, the fruit pomegranate, came up. So pomegranate is a fruit that is of medical importance because it does act as an antioxidant. It helps with inflammation and it also reduces, it's a toxin reducer. So pomegranate juice is also uh, believed to improve neuroprotective features against oxidative damage, and it also enhances neuron survival. So, these two researchers that conducted this specific study had done a study similar in the past with pomegranate juice and its oral administration. So, their data from the previous study revealed that the pomegranate juice did alleviate developmental and biochemical alterations in the brain, so helping with learning and memory impairment as well as sexual behavior dysfunction after the mice were exposed to aluminum trichloride. However, they were unsure whether oral treatment of the pomegranate juice could moderate the risks of depression and anxiety in aluminum trichloride treated mice. So, therefore, this study was aimed to explore the effects of pomegranate juice on anxiety and depression like behaviors induced by the exposure of aluminum trichloride in male mice. So, before they began their study, they did hypothesize that pomegranate juice would reduce the effects of anxiety and depression like behaviors caused by aluminum trichloride. To test their hypothesis, they collected lots of mice that were approximately 11 weeks old and weighed 30 grams. They then split these mice off into 6 different groups and had them together in their assigned boxes. So the first group was the control group that was not exposed to anything. They just received tap water. The second group was given a concentration of 20% of the pomegranate juice. The third group received a 40% concentration of the pomegranate juice. and the fourth group received 400 milligrams of the aluminum trichloride. And then the last two groups received the aluminum trichloride along with the 20% and 40% of the concentration of the juice, respectively. So this treatment and the different exposures went on for 35 days. And then after those 35 days ended, they looked at the behavioral assessment or they did a behavioral assessment on the mice. So for this assessment, they did 5 different tests. The first test was the elevated plus maze test. The second test was the open field test. Then we had the forced swimming test, the tail suspension test, and the light and dark box test. After the mice went through all of these tests, they then did a brain biochemistry analysis, and they specifically looked at the levels of cortisol, which is the body's main stress hormone, and they were just trying to see if it would increase or decrease depending on the different exposure that the mice received. So now I'm going to get into the different behavioral assessments, and I'm just going to give a little bit of background of them and how they worked, what they did, what the expected results were, and what they did conclude after doing the different assessments on the mice. So the first one is the elevated plus mace test that is commonly used for anxiety research in rodents. So if you look at this picture that I have on the left, this is kind of the setup that they had. They had a maze that contained 2 open arms and 2 wall-sheltered arms. And this was done to kind of analyze the mice's tendency to stay in an enclosed space. So typically they refer to the normal mice or the mice that weren't exposed to anything as moving freely and very curious and just being in open spaces. But they did predict that the mice that were exposed to the aluminum trichloride, so the depressed and anxious mice, they would spend more time in enclosed spaces. So after doing the assessment on all of the mice, they did conclude that the aluminum trichloride exposed mice spent more time inside of the wall sheltered arms and in the middle space, so where the two different arms meet. The open field test is one of the most used tests to measure behavior in animal models, specifically looking at the willingness to explore. So typically mice are very curious, are always willing to explore, just familiarizing themselves with their surroundings. So to do this, they had a square clear box, and they had a camera over it so that they were able to record the mice and then look at the recordings later. So they had 2 groups of the emitter and sensor arrays for both the horizontal and vertical activities. And they expected that the mice with increased anxiety would have less locomotion, so therefore not really move around a lot, and they would have less of an interest of exploring their surroundings. So from this assessment, they were able to detect that those exposed to the aluminum trichloride had more of an anxious response, so therefore not really moving around. For the forced swimming test, they did have a tank that was 30 centimeters in diameter. So what they did is they filled the tank with tap water set to a height of 20 centimeters, so only leaving about 10 centimeters or so free for the mice to be able to poke their heads up for some air. And the water was replaced after every mouse was tested. So the mice that were unexposed were expected to move around as soon as they were dipped into the water, just to see how they were expected to move around in the water just because that's their normal behavior. So when they did this study, or when they did this test, they looked at the different sessions by recording them, and then they looked at the frequency of the immobility, swimming, and any struggling for the 2-minute mark, 4-minute mark, and the 6-minute mark. So kind of similar to the last test, they did expect that the depressed mice would be immobile, so not moving, not really having much of an interest, and just kind of chilling in the water, right? So when they did this, they discovered that the aluminum trichloride exposed to mice that had depression-like behavior, they had an increase in time and in the frequency and immobility parameters, and they had less time in swimming and climbing parameters compared to the unexposed mice. For the tail suspension test, they hung the mice upside down, and they suspended them by the tail at 40 centimeters using sticky tape. So again, this is used to measure the stress in rodents. The mice that were stressed would be immobile and they wouldn't really do anything, whereas the mice that were not stressed or not exposed to the aluminum trichloride were expected to move around and trying to set themselves free. So after doing this, they did notice that the aluminum trichloride exposed mice showed depression-like behavior by increasing immobility after looking at the 2-minute, 4-minute, and 6-minute mark after the suspension. And then lastly, the last test that they did was the light and dark box test. So they had 2 different chambers. The light chamber contained a bulb of 60 watts, and the dark chamber was completely dark. It had black walls and it was covered with a black roof. So what they did is they placed the mice into the light chamber first, because typically the mice like to be in the light and they're always very curious and moving around. So they left the mice, or they left the mouse because they were doing it individually. Each mouse was placed in the box into the light chamber and they were left in there for 5 minutes to do as they pleased. And they expected that the control group would be attracted to the light chamber just due to their natural aversion to brightly illuminated areas, and as well as, that curious behavior or just getting familiar to their surroundings. So what they found is the mice exposed to the aluminum trichloride showed anxiety behavior because they were attracted to the dark chamber. So they went from the light into the dark and they stayed there for most of the time. So the graph that I included here, on the x-axis, we have all of our groups. We have the control group, the 20% concentration, 40% concentration, the aluminum trichloride exposed group, and then the 2 groups that received both the aluminum trichloride and then the 2 different concentrations of the aluminum trichloride, sorry. And the y-axis is just looking at the cortisol levels. So this is the biochemical analysis that I was talking about after they did the behavioral assessment. So what they found is that the aluminum trichloride exposed group had the highest level of the cortisol, meaning that they were the most stressed. And then following them was the group that was exposed or that received the 40% concentration of the pomegranate juice. And it was very surprising to learn that the group that had the lowest level for the cortisol, which is the stress hormone in the body, was the group that was exposed to the 20% concentration of the pomegranate juice. So from that, we can gather that the hypothesis was supported because the anxiety and depression-like behaviors induced by the aluminum trichloride exposure can be improved by the pomegranate juice. And this was seen with, looking at the cortisol levels and all of the behavioral assessments. All of the mice pretty much reported or behaved in the same way, and the control group was completely different to that of the mice that were exposed to the metal. So from that, the main takeaways that I want to leave you all with is that it is being discovered that some metals are being linked to many neurological pathologies, and that exposure to aluminum is inevitable because it is found anywhere. So therefore, being exposed to that aluminum trichloride leads to the neurodegeneration in the brain, which is then associated with depression and anxiety, which is what was occurring with the mice. Also, just that the pomegranate is very useful, very healthy, and it is of medical importance, specifically for the mice. So the mice that were receiving the pomegranate juice uh, were kind of taking those negative effects away and just moderating it a little bit better and making it so that they weren't as immobile or sticking to the dark zone how it was with the light and dark test and all of the other behavioral assessments. So yeah, that also goes with the last point, that oral ingestion of the pomegranate juice moderates the risks of depression and anxiety caused by the aluminum trichloride. So that is it, and thank you so much for watching.